Hello, everybody. Thank you for joining me today. My name is Julian Kellner-Rogers, and today I'll be presenting my senior thesis research on chemical inhibition of the quorum sensing signal synthase. Okay, so first I'd like to give you a little background into the quorum sensing system. So I like to describe quorum sensing as this unique ability that some bacteria have to communicate with one another and turn specific behaviors on and off. And they do so via the production of these small signaling molecules called autoinducers, which are pictured in these little reaction flasks as the yellow circles. And so how it works is when the bacteria are low in concentration, the concentration of autoinducers will also be low, which results in the target behavior being turned off. As the bacteria grow in concentration, so too does the concentration of the autoinducers, which results in the target behavior being turned on. So in the example we have here, which is Chromobacterium violaceum, it undergoes this change in pigmentation from this pale yellow color when the autoinducers are low in concentration to this nice violet color when the autoinducers are high in concentration. So here we'll be looking at the quorum sensing mechanism. And so how it works is this enzyme called the autoinducer synthase catalyzes the production of these small signaling molecules called autoinducers, which are pictured here in yellow. Once the autoinducers reach a threshold concentration, they then bind to the autoinducer receptor, which in turn binds to the DNA, leading to transcription of target genes or the turning on of the desired behavior. So if we think back to our example of Chromobacterium violaceum, this would cause that change in pigmentation from yellow to violet. And on the next slide, I'll show you, we actually have a nice X-ray crystal structure of this complex pictured here. Okay, so as promised, pictured here on the left, we have this nice X-ray crystal structure. And so if you look closely at this gray ball and stick model here, that is actually an autoinducer, which is bound to the autoinducer receptor, which is pictured in blue, which is bound to the DNA in yellow. And then over on the right-hand side here, we just actually have some structures of autoinducers. And so it's important to note that in our lab, the enzymes we work with catalyze the production of autoinducers that are defined as acyl homoserine lactones. And so all of the autoinducers that we work with are going to have this identical homoserine lactone group that you can see in all of them. And so the only ways that these autoinducers differ are in the length of the acyl tails as well as the oxidation state at the third carbon. Okay, so the question is, why do we care about all of this? So let's look at the example of Pseudomonas aeruginosa. Pseudomonas aeruginosa is an opportunistic human pathogen that is known for forming these antibiotic-resistant biofilms, and it often affects immunocompromised patients and patients with cystic fibrosis, where it can colonize in the lungs and is very often fatal. And so what's important to note about Pseudomonas aeruginosa is that the expression of its virulence genes requires cell-to-cell communication, or quorum sensing. So you could imagine that if we could stop Pseudomonas aeruginosa from quorum sensing, it would no longer be able to act as a human pathogen. So the goal of my work is to synthesize transition state analogs to chemically inhibit the autoinducer synthase enzyme. If we can inhibit the autoinducer synthase, it will no longer produce autoinducers. They can't bind to the autoinducer receptor and will no longer get transcription of the target genes. So thinking back to the case of Pseudomonas aeruginosa, it would no longer be able to form those antibiotic-resistant biofilms. And this could kind of be thought of as a new type of antibacterial strategy. In this slide, we'll be taking a closer look at the mechanism through which autoinducers are produced. So the two substrates are this acylated acyl carrier protein and SAM, which stands for S-adenosylmethionine. They react in the presence of the autoinducer synthase to form the autoinducer as well as MTA, methylthioadenosine. So looking down at the bottom here, we have the mechanism. So the reaction is initiated by this nucleophilic acyl substitution reaction between S-adenosylmethionine and acyl ACP. We then get this intramolecular ring closing reaction which produces the autoinducer and methylthioadenosine MTA. Here we'll be taking a closer look at the assay we use to detect enzymatic activity. So it's important to note that when acyl ACP and SAM react to form the reaction intermediate that we saw on the previous slide, they also produce this thio-ACP as a byproduct. And so our assay involves the use of this small molecule called DCPIP. DCPIP is a thiol detecting reagent, and so how it works is DCPIP is naturally blue in color, and it gets reduced in the presence of thiols and becomes colorless. So our assay consists of adding the substrates, both acyl ACP and SAM, as well as the autoinducer synthase, into a 96-well plate with this small molecule called DCPIP. We then monitor its absorbance at 600 nanometers, which is the wavelength that blue light absorbs at. And so if we are seeing enzymatic activity, we would also expect that this thiol is being produced, and therefore DCPIP should be getting reduced from blue to colorless. And so if we look at the right here at the graph that we have, we see exactly that. So both our no enzyme and heat-treated controls, we can see that the slope is completely flat, meaning that the DCPIP remain blue, no enzymatic activity. And in both the samples containing enzyme, we see this steep reduction in slope, indicating that this thiol is being produced, and hopefully we're seeing enzymatic activity. So this brings us to our first inhibitor, JKR. So JKR was synthesized using our knowledge of the reaction intermediate, as well as by using X-ray crystal structures of the autoinducer synthase enzymes. So if we look at this reaction intermediate, which is essentially the closest thing we have to a transition state, we can see that JKR is essentially identical to this reaction intermediate. The only real difference that you could maybe spot is there's this one extra carbon here. And then if you look over to the right at this X-ray crystal structure, this is methylthioadenosine bound to an autoinducer synthase. And this structure shows us two things. One is the importance of the adenine ring. This shows us that the adenine ring makes hydrogen bonds to the autoinducer synthase, which is important for holding it in place, which is why we included this adenine in JKR. The other thing that we can learn from this X-ray crystal structure is that the stereochemistry is very important. You can see that we need cis stereochemistry to guide the acyl tail into this pocket in the active site of the enzyme. And so that is why if you look at this lactone ring on JKR, you can see the cis stereochemistry here, which was designed to help guide this acyl tail into the active site. Here I'll be taking you through the synthesis of JKR. So it starts with L-allylglycine, and in the first step we just Bach protect the amine. We then take this Bach protected L-allylglycine and react it with NBS, which results in the formation of the bromonium ion at the alkene, followed by a nucleophilic attack of the hydroxyl group to form the lactone ring. We then deprotect the amine and couple it with octanoyl chloride to form this acylated homoserine lactone. We then react the acyl homoserine lactone with this protected thioadenosine in the presence of sodium methoxide. The sodium methoxide cleaves this thioester bond, which frees the sulfide, resulting in a SN2 reaction between the sulfide and this alkyl halide. This forms acetal-protected JKR. We then deprotect in weak acid and water, which gives us JKR. After synthesizing JKR, we tested its activity using our DCPIP-based assay against the autoinducer synthase enzymes BMI1 and CEPI. And so it's important to note that both BMI1 and CEPI catalyze the production of autoinducers that feature an 8-carbon acyl tail. This is why JKR also features this 8-carbon acyl tail to closely match the structure of the autoinducer that's being produced. So we tested JKR at concentrations of 1, 10, and 100 micromolar. And so against BMI1, we did see some activity. We found that it had an IC50 around 100 micromolar, but we actually found that it was much more potent against CEPI. And so against CEPI, the IC50 is right around 1 micromolar, And at both the 10 and 100 micromolar concentrations, we essentially see complete inhibition. And this is actually much more potent than many previously reported inhibitors. So if you can remember back to the beginning of the presentation, I said that all of the enzymes that we work with catalyze the production of autoinducers that are acyl homoserine lactones. And so they only vary in the lengths of their acyl tails. So on the previous slide that we just looked at, we had BMI1 and CEPI, which both catalyze the production of an autoinducer with an 8-carbon acyl tail. Here we're looking at the synthase REL-I, which catalyzes the production of an autoinducer with this 4-carbon acyl tail. So we reasoned that if we modify the acyl tail length on JKR to more closely match the structure of the autoinducer that is being produced, JKR will have a better fit into the active site of the enzyme, and we may see better inhibition. So to test this, we modified the length of the acyl tail in JKR to feature a 4-carbon acyl tail, which we call butyryl JKR. We tested both butyryl JKR and our original JKR using our DCPIP-based assay against the autoinducer synthase REL-I. And again, REL-I catalyzes the production of an autoinducer that features a 4-carbon acyl tail. So we tested both compounds at a concentration of 100 micromolar, and unfortunately we didn't really see any activity from either. If you look at the 100 micromolar concentration of our butyryl JKR here, you maybe see a little activity, but again, 100 micromolar concentration is very high and essentially no activity, so this wasn't very promising. Now, there could be a couple of reasons this happened. We already know that JKR isn't active against every single autoinducer synthase enzyme that we've tested it against. And additionally, there is a loss of hydrophobicity that comes with shortening the length of the acyl tail, and that could lead to decreased interactions between our butyryl JKR and the active site of the enzyme. Another modification we made to JKR was at the lactone ring. So when we were looking at crystal structures of some of these autoinducer synthase enzymes, we didn't really see any real interaction between the lactone ring and the active site of the protein. So we wanted to come up with a method to determine how important this lactone ring moiety is to our JKR compound. So to do that, we just replaced the lactone ring with this simple 5-carbon alkyl chain for a molecule that we call linear JKR. Here I'll be taking you through the linear JKR synthesis. So we start with this 5-hydroxy-1-pentylamine, and that gets heated under reflux in the presence of HBr, which converts this hydroxyl group to the alkyl halide that we see here. This is then coupled to octanoic acid using the coupling reagent EDC. And then the resulting compound is then coupled with this protected thioadenosine here in the presence of sodium methoxide. We've actually seen this reaction before in the original JKR synthesis. So the sodium methoxide cleaves this thioester bond, frees the sulfide, which enables it to undergo the SN2 reaction with the alkyl halide. This results in an acetal-protected linear JKR. We then deprotect the acetal using formic acid and water, which gives us our linear JKR. We then tested the activity of linear JKR using our DCPIP-based assay against the autoinducer synthase CEPI. Now, if you remember, CEPI is the synthase which the original JKR showed the most activity against. So we tested linear JKR at concentrations of 1, 10, and 100 micromolar, and we also used a 100 micromolar concentration of JKR as a positive control, which is shown here. And what we found was pretty interesting. So we found that linear JKR did show some activity against CEPI, but that it was independent of the concentration that we used. And so what this means, if we look over here, the 1 micromolar concentration showed more activity than the 10 micromolar concentration, and then the 100 micromolar concentration showed about the same activity as the 1 micromolar concentration. So it's entirely possible that this is just due to human error and maybe I messed up the assay. And before we can draw any real conclusion from this, we should probably repeat the experiment. However, that hasn't really been possible due to the recent pandemic. Now we'll be getting into some projects that I had just recently started working on, but unfortunately wasn't able to make much progress on before everything was shut down. And so they turn into future directions, and hopefully someone else can work on them in the future. So what we're looking at over here on the left is an X-ray crystal structure of an autoinducer synthase enzyme. And what we can see here circled in red, this is actually a non-catalytic cysteine residue that's found in many of the autoinducer synthase enzymes that we work with. And these non-catalytic cysteine residues are located deep within the acyl pocket in the active site of the protein. And so we reason that if we modify the end of the acyl tail in JKR to be fit with various electrophiles, these electrophiles would be susceptible to nucleophilic attack from the thiol present in the cysteine and could potentially covalently inhibit the enzyme. So the two ideas that we had were, one, an isothiocyanate, and two, a Michael acceptor. Molecules containing both a Michael acceptor and an isothiocyanate have been shown to be effective at covalently inhibiting enzymes and even some quorum sensing enzymes. So now I'll be taking you through the proposed chemistry for the isothiocyanate JKR electrophile. So we start with this 6-bromohexanoic acid. It'll undergo an SN2 reaction with sodium azide, which gives us the 6-azidohexanoic acid. That is then coupled to 5-bromo-1-pentylamine using the coupling reagent EDC, which gives us the resulting amide. That compound is then coupled to this protected thioadenosine. We've seen this before, SN2. Great. This resulting compound is then reacted with carbon disulfide and triphenylphosphine, which converts the azide into the isothiocyanate. From there, it's just a simple acetal deprotection using formic acid and water to convert the acetal to the diol. So here we have the isothiocyanate linear JKR version of the compound. It's entirely possible that if we wanted to make the original JKR with the isothiocyanate, we could do that. We just have to change the chemistry earlier on. Now I'll be taking you through the proposed synthesis for the Michael acceptor version of JKR. So it starts with the reaction between tetravinyl tin and N-butyl lithium, which produces vinyl lithium. Vinyl lithium then reacts with glutaric anhydride to produce this 6-oxo-octenoic acid compound. And don't get confused because we actually don't use this compound directly in the next step, but it comes into play later. So we then take this Bach-protected 5-bromo-1-pentylamine and couple it to our protected thioadenosine, as we've seen before with the SN2. That resulting compound is then deprotected using trifluoroacetic acid, and the amine is coupled to the oxo-octenoic acid compound that we previously made. We then just have to deprotect the acetal group, seen here, using formic acid and water, and that gives us the Michael acceptor version of linear JKR. Again, just like before, if we wanted to make the Michael acceptor version of the original JKR with the lactone ring, we could probably do that. We just have to change the chemistry a little. So before we end, I just wanted to take you through one last idea we had that we never really were able to test, and that is to synthesize JKR analogs by replacing the lactone ring present in JKR with naturally occurring alpha amino acids. And so if you can visualize this, replacing the lactone ring with the alpha amino acid is essentially acting like a ring open version of the lactone ring. Additionally, it allows us to make a wide variety of new molecules just by varying the R groups present here. So say we wanted this to be a hydrogen, we could start with glycine. If we wanted it to be a methyl group, we could use alanine, and so on and so forth. And the other thing that's cool about this is the stereochemistry of naturally occurring amino acids matches the stereochemistry that's present in JKR. Okay, thank you everyone so much for watching my talk. I can't believe this is the end of my senior year. So I, of course, have to thank Scott for everything, working with me every day, helping me on this project, everything. Julia Stockwell for always keeping me entertained and making me laugh. Nora and Andy for annoying me every day. Everyone, all the professors that I see, it has been such a great experience, and I'm definitely going to miss you guys. I'm really sad to be leaving. I'm also really excited to start my PhD, and I'll only be like 15 minutes away, so hopefully I can come visit you as often as possible. Thanks so much, everyone. Bye.